everybody. My name is David. I go to Cypress College and my major is chemical engineering.
Hi, I'm Numa. I go to Cypress and I'm majoring in data science.
Hi, I'm Andrew and I'm upcoming senior at Baltimore High School.
And our project was on particle tracking, velocimetry, and an Acousto-Electric Microfluidic device. Intro.
So the long-term goal of our experiment was to find the most effective way of intracellular delivery through the device called ASOC, or Acoustic-Electric Shear Orbiting Platform. So intracellular delivery is a process of inserting molecules, which we call as cargo, into cells. And this is done in a way in which all cells in a sample receive the cargo. And this can be done either through a carrier meditated way on the right side, so through nanocarriers or viral molecules, or it can be done through direct membrane penetration, which is the way our device uses. So the reason why intracellular delivery is really important is because it provides researchers and other people in the field a way to edit genes, improve disease treatments, and also improve cell fate, function, and behavior.
And for the ASOC device, this is the device that allowed us to, track the cells and look underneath the microscope. And as you can see in the top, it's the chip that we used underneath the microscope. And what we did, we had an input and an output, circles that we input them in one, and then they would flow through the air-liquid interface, and they would go through these vortexes that would allow them to be sheared, opening up their pores, allowing for the cargo to enter in the membranes. And this was the PZT that allowed for the electricity and to go through and activate the oscillators to move through it. And overall, the system allows for shearing to occur, opening nanopores in cells to allow cargo to go through and into it.
So in comparison with other methods to create intercellular delivery, Nura mentioned viral delivery, viral vectors. Although unfortunately, with this specific technology, this has to be done in a clean room. It has to be done very carefully. By virus, I literally do mean like the viruses that get us sick. Instead of having normal DNA there that would usually infect us with the cold or the common flu, there would be the package of mRNA or ssRNA in there. With electroporation, there is an extremely hard electrical circuit that runs through the cell and opens up these pores. However, there's no way to really control the electricity and the amount that it opens, so it can have dependencies in the transfection efficiency and also some risk about the cell being able to recover and work again. And finally, with lipofaction, this is in reference to the other delivery, similar to ribosomes in the cell that carry and transcribe and transcript. They made artificial liposomes that go into the cell in a very similar way. Although again, for this, the viability for the cell is moderate and the transfection is also technical. For the ASOC specifically, by using this piezo that just vibrates at a specific harmonic frequency, we're able to create those vortexes that create their own pump. We don't need to use an external pump to get the cells through and cause the shearing mechanism. So it's extremely cheap. There's no need for clean rooms or expensive equipment as much, and it's very easy and viable to do.
And the methods that we used over the week. So for the first part, we did the 2D T-cell GearCat particle tracking. So in the top, it's kind of like the LCT that we used throughout the system, and these are the PZT, the side ones, allowing for the circuits to go through. And we have the different dimensions. So this top one was a 500 by 60 micro dimensions, and the bottom ones were smaller, narrower, and wider ones. And the big photo shows tracking these cells through these pathways. And you can't see it now because we don't have it, but the side charting with the colors show the velocities that these cells were going in.
So in order for us to actually view the motion that's happening inside of the chip in 3D, we have to use the lattice sim. And instead of using the cells inside of the chip under the lattice sim, we used fluorescent beads. And the reason we used the beads instead of the cells was because the beads were 10 times smaller, so they were only 1 micron. And also the fluorescence allowed us to view it a little bit easier. So we inserted the beads into the chip, and we put the chip under the lattice sim. And what the lattice sim did was it took a z-stack, so it allowed us to see the direction of the cell movement in the z-axis. So on the right side is a picture of just one piece of the z-stack, I guess. So that's just showing x and y movement. But once you have all the slices of the z-stack, you can see movement across the z-axis as well.
And our results.
So this was a graph that was created by TrackTrack, which is an open source code that was created by a professor in Columbia. I'll take you one step at a time. So here, this is the U vector and V vector, or more commonly x-axis, y-axis, right? So these colors here correspond to the movement depending on those axes. So here, if I were to draw a plane, right, like this would be the x-axis on the positive side here, and negative here. On the positive side, the velocity going toward the positive direction here, the stronger it is, the more red it is. So we can tell that these cells are moving toward this direction, toward this little liquid air interface that's allowing these vortexes to form. So then we can see that it's making this general circle, and then over here it's the same. So they would travel in these directions. And then from the y-axis as well, this will be up and down movement. So we can tell here that the cells are moving upwards here, the velocity is going up, and then here the velocity is going down. My colleagues here, this is the control that we had. This is the typical one they used in the lab, which is 500 microns large. We also had ones we tested with 250 microns and 750 microns.
So the 250 micron is the smallest chip that we use. So here you can see that in the smallest channel we had the smallest vortices, and the cells were a little bit closer together. And this created a more uniform velocity, which we assume would be more uniform shearing, or a more uniform nanopores formed across all the cells. And uniformity is what we're trying to strive for in the different chips that we test, so that all cells receive correctly.
And we also tested the 750, which does quite the opposite of the 250. Just because the channels are much wider, the vortices are much larger, and as you can see, they have this the middle split, which was not on the 250, and it doesn't really allow for as uniform as the 250 or the 500. Exactly.
So with some of our 3D products as well, I should specify that we had two different experiments going on, right? So that was one with the cells. This is the one that we had earlier with the fluorescent beads. Can you play the video?
Yeah.
Here, our computers crashed, I think about 6 times while we were doing this, and Adila knows that we were struggling a lot to get the particle tracking on here, but this is very novel actually. Within the Biomedical lab, they actually haven't they've proven before that there are vortices in the z-axis, but they haven't seen it, and this particle tracking through a mirror was able to prove that there is movement between both the x, y, and the z-axis altogether. So in conclusion, for the 2D particle tracking, we were able to determine in general, the smaller the channel width in which the vortices are being created for the cells, the more uniform the cell velocity, which means in general, there will be more uniform shear. It's been theorized, though it hasn't been proven, that with more uniform shear, then every single cell would have approximately the same amount of nanopores, and it's about number, not size, if I remember correctly, to allow the mRNA in. Although it is very hard, I wish we had a second week, it's very hard to see the nanopores specifically because they can close in 3 to 4 seconds. So it's very hard to get them under the microscope. And then for the 3D particle imaging, again, it was just a proof of concept. This is the first time that they were actually able to see the 3D vortices moving in the x-axis, and yeah, that was new to the lab. But after all that, I'm sure some of you are wondering, okay, you put cells and beads into a little washing machine. Now, what does it do? So in terms of application, this can actually be used to treat cancer. With chimeric antigen receptors, or CAR T cells, they're created by creating this, having a cargo of mRNA or ssRNA to be put into the T cells, right? And it's specifically for the ASOP, for this device, it's able to control frequency, right? Which just means it's able to control the amount of voltage and the overall effect, the amount of mRNA that the T cell itself is able to receive. So in this case, cytokines here, they're similar to like little highlighters or little alerts for the immune system to start attacking things. So in this case, if we have just enough, then it will be able to attack specifically cancer cells or different types of diseases. While if we use that other process I mentioned earlier with electroporation, we're unable to control the dosage and the voltage, which means that there could either be too much, which causes extreme inflammation in something called cytokine release syndrome, or too little, which would not be able to resolve the disease or the immunogenicity at hand. And I would like to acknowledge Manor for sitting with us through all those computer crashes and everything that we had to go through. Professor Abraham Lee, who runs the Biomedical lab, and Professor Dignan for all of her work in the US IVR and for treating us to such great breakfasts and lunches. And also, a special thanks to Adila. I know she was a microscope superstar this week. I know she's been working for all the groups, as well as Mohammed, who helped us figure out the programs. Thank you so much.